Okay, ladies, so here is a lecture to go along with Chapter 9 that you have read or will be reading soon, which covers the cell cycle, so taking us back to mitosis, as well as how we control the cell cycle and some little aspects of cancer as well. Traditionally, we did this talk in class last year. It didn't take that long really in class, but it did really shortchange us in terms of time to do the lab on mitosis. So this way I thought we could flip our classroom, you could listen to this on your own time, and then that would give us a full class time to do this mitosis lab coming up. So this is also the first time I've done a lecture with Samson sitting at my feet. So there's always something new and fun, but we're going to talk about how your cells divide. We're going to talk about how we regulate that whole process, and we're going to talk just a little bit about cancer as well. Now, pretty much, in terms of mitosis, you guys are on your own and understanding the cell cycle. I think you've had this a couple times now, probably 7th or 8th grade, and then I know you had it from me as freshmen, and I'm really confident in your all's ability to handle that stuff on your own. A few things to remember. Don't forget that the cell cycle is really 2 steps. It has a mitotic or M phase, which takes turns with interphase. And if we drew this like a pie chart, mitosis, or the M phase, is really tiny, and interphase is going to take up the rest of this. So at any given moment, most of the cells in your body are going to be in interphase. Don't forget the interphase we break into G1, S, and G2 stages. These are primarily growth stages for the cell, and then the S would stand for synthesis, which is synthesis of DNA, right? So DNA replication, which we'll spend a lot of time with later this year, is going to take place during the S stage of interphase. Now we also have the M phase, or mitotic phase. You probably remember those stages that go along with that, starting with prophase. Then a lot of texts and colleges talk about late prophase and pro-metaphase. We lumped that into prophase when you were a freshman, and we still can kind of think of it that way. But after that, we've got metaphase, anaphase, and then telophase. One thing to point out, make sure you realize, is that cytokinesis is a separate thing all its own. It's actually a separate step outside of mitosis. And that actual splitting of the cell is considered its own thing. So M phase consists of mitosis and cytokinesis, which are technically 2 different processes. Now I went ahead and included some pictures that I like of these steps in the cell, starting out here with what an interphase cell would look like, followed by prophase. Remember, in prophase we're going to form the spindle, we're going to break down the nuclear envelope, we're going to see those chromosomes condense into an X-like shape with 2 sister chromatids in each of those X's. Then the chromosomes are going to align themselves somehow on the spindle, and that's what we often say takes place in this late prophase or pro-metaphase. Then we've got metaphase. Don't forget, M, think midline, so the chromosomes line up on the midline. Anaphase, remember A, think apart, so the chromosomes are going to spread apart into 2 groups. And then telophase would be where our nuclear envelope reforms around each set of chromosomes. Nucleolus returns, spindle sort of breaks down a little bit, and then cytokinesis would be the actual cell splitting, and that takes place along what we call the cleavage furrow, if we're looking at an animal cell that doesn't have a cell wall. Some other key terms to note from the early part of the chapter, eukaryotic cell division is going to give you a particular result, and that result is 2 cells which are genetically identical to one another. So remember that when it comes time to compare mitosis to meiosis. Mitosis is going to give us 2 cells that are genetically identical. Now these cells, for most of the parts of your body, are going to be what we call somatic cells. Somatic is just another term for your general body cells, so skin cells, heart muscle cells, liver cells, you name it, those are somatic. And all the somatic cells in a human are diploid, or 2N, meaning that they have a double set of chromosomes. Basically, their chromosomes come 2 at a time in pairs, and each of those pairs is a grouping of one chromosome that you got from your mom and one chromosome that you got from your dad. Now we also have other organisms that can be what we call haploid, represented by just N. In a haploid organism, the cells only have one set of chromosomes, so they don't come in pairs. For humans, we've got two specific cells that are haploid. In males, we have sperm, and in females, eggs. So the haploid cells of the human are the reproductive cells, and there's a reason for that, right? If each of these has half the number of chromosomes, then when we have sexual reproduction, we end up with a full set of chromosomes in our kids, and so that's why we need those haploid cells. Now prokaryotes, a little bit different story. Their cell cycle is not nearly as well controlled as ours, and they undergo what's called binary fission, which basically just means to split into two, right? This is not nearly as a complex a process as we see in eukaryotic mitosis. Other major key terms that you're going to want to make sure that you know by test time are on this slide. Check these out in the book if you're confused at all by this. Notice here in animals we have a cleavage furrow, so the membrane just kind of gets pinched off. In plants, we form what's called a cell plate. So we actually, if we have a plant cell here with a cell wall, we're going to build a new cell wall there to turn it into two cells, and that's what we call a cell plate. Also apoptosis, or programmed cell death, we're going to talk about much more here in the coming days, but you might remember that term as cell suicide from previous biology class. Now, chromatin packaging is one thing that I want to talk about for a moment that was in this chapter, because I want to make sure that you realize that DNA is not just hanging out willy-nilly in the cell. It is packaged in a very specific way with proteins within the cell. And so at the smallest level we've got DNA, but then DNA is going to wrap around proteins called histones. So each of these little blue spools in this picture is a histone. And when we wrap the DNA around the histone, DNA here in red, histone in blue, we get what's called a nucleosome. So each of these little structures with the DNA around them is called a nucleosome, oftentimes referred to as beads on a string when we look at it under an electron microscope. The next thing your cell does is it kind of twists those nucleosomes around one another to form a fiber called chromatin. And so chromatin is a DNA plus protein fiber that we see inside every cell's nucleus. Chromatin just sort of gets aligned on a protein scaffold, but really it just kind of bundles up inside the nucleus. And this is what we see in a normal cell. So a normal interphase cell is going to have just this bundle of gunk basically in the nucleus, which is its chromatin. However, as we approach mitosis, so late in interphase, we will see that chromatin condense into what is actually a chromosome. Now chromosomes generally take on this X-like shape, where each side of the X is one of the sister chromatids, and this is basically just like you wadded up a string. So if you ripped a little string off your shirt one day and then you just rolled it between your fingers and wadded it up, that's basically what's happening as chromatin becomes a chromosome, is we're just going to wad up that little string that we have in there. So I do want to spend more time with how your cells control cell division. This is something that there are usually several questions on the AP exam about, and so something that I didn't want to just leave to your reading. A eukaryotic cell has a really specific control system that is very, very finely tuned and occurs at a tiny, tiny molecular level. But this control system differs for every type of cell in your body. Some cells divide a lot, like your skin cells. Right? You guys know you constantly lose skin cells, and so those cells are going to constantly be dividing and replacing themselves. Other cells never divide, like a neuron. Once you have a nerve cell, it doesn't do cell division. There's been major research done trying to figure out how to get those nerve cells to actually divide so that we could heal brain injuries or spinal cord injuries, and it just doesn't happen. Then you've got sort of a middle ground. You've got cells like liver cells that are capable of dividing, but that usually don't. And so all of this has to do with various controls that are going on in those cells. If we go back to the 1970s, there were tons of experiments that happened to try to figure out how a cell controls itself. And the most successful experiments took a cell that was actively dividing and fused it with a cell that was not dividing. We'll make him sad. When we did this, what we saw is that the second cell would start to divide. So we'll have him grow in a new cell here or something. What that told us is that something in the cytoplasm is probably causing cell division to take place. Some sort of cytoplasmic signaling. If the cytoplasm of this first cell introduced to the second cell will cause that second cell to divide. This suggests that you almost have an internal clock like a washing machine. Right? A washing machine, you set it and you forget it, but it has a system there and it's going to run for so long. We think a cell has a similar system inside where it's got a clock and it understands how long something has been taking place and it helps control that. In a cell, we refer to a control point as a checkpoint. So any point in the cycle where the cell is told to either stop dividing or to go ahead and continue dividing, we refer to as a checkpoint. And if you take a look at this picture here on the upper right, you see the cell cycle, again, the really short M phase and this long interphase, and you notice that there's three major checkpoints. There's a G1 checkpoint, which takes place toward the end of G1. Then there's a G2 checkpoint, which takes place right at the end of G2, and an M checkpoint, which happens right in the middle of mitosis. Now the major checkpoints, like we just said, are at G1, G2, and M. G1 is probably the most important checkpoint, and we refer to this as the restriction point. If a cell gets a green light, like we see here, during G1, it's probably going to totally divide. So it's probably going to go all the way through the cycle from there. And the other checkpoints are probably not going to stop it either. So if a cell goes ahead through the restriction point, we probably see cell division in its entirety. However, if a cell gets stopped at the restriction point, then the cell moves into what we call G0, or sometimes G0. Right? This G0 phase is a non-dividing phase, which means the cell is not actively undergoing cell division. Don't be confused here that the cell's alive. It's not dead or anything like that. It's just not active.